Total maximum daily loads, TMDLs, are the tool used by environmental regulators to address non-point source pollution under the Clean Water Act. The Act's policies and goals are that it is the national policy that programs for the control of non-point sources of pollution be developed and implemented in an expeditious manner so as to enable the goals of this Act to be met through the control of both point and non-point sources of pollution. When non-point sources of contaminants and pollution adversely impact specific designated uses, the regulators must assess water quality status before a corrective TMDL is needed. A total maximum daily load is the quantity of pollutant determined by the regulatory agency to be the greatest amount a defined water body's assessment unit can absorb each day without adversely impacting a designated use. The agency partitions this quantity into waste load allocations for point source dischargers and load allocations for non-point source dischargers. Assessment units can be a stream or river reach, a tributary, subbasin, or entire river network. How an assessment unit is analyzed is critical to properly identifying designated use status. It should be specific to a designated use in that assessment area and a specific pollutant. Regulators often struggle with water body assessment, and it's common for affected industries in the regulated community to be dissatisfied with the results. In this tutorial, I present the history of water quality protection and TMDLs under the Clean Water Act and explain why regulators find it difficult to quantify ambient conditions relative to designated use standards. These issues make it difficult to determine a TMDL and effectively allocate the discharge limits to point and non-point dischargers. Recommendations for increased effectiveness of the water body assessment process by incorporating appropriate environmental science and data analyses were developed in 2001 by the National Research Council in response to Congress's demand that they analyze on a broad scale the scientific basis of the TMDL program. Not all states or the EPA have implemented all their recommendations. Federal action to protect the nation's waters dates from the Rivers and Harbors Appropriation Act of 1899. In 1948, the Water Pollution Control Act was passed by Congress. Both laws used ambient conditions to determine water quality in an assessment unit. 24 years later, the Water Pollution Control Act Amendment of 1972, the CWA, separated point and non-point sources and focused on the former using effluent-based water quality standards. This act was supplemented by the Clean Water Act of 1977 and the Water Quality Act of 1987, which restored ambient conditions to set water quality standards. These three laws are the current drivers for protecting the nation's water resources. The focus of water quality laws was originally on human health. This focus gradually broadened to include aesthetic uses such as fishable and swimmable. The 1972 CWA expanded the goal to, quote, restore and maintain the chemical, physical, and biological integrity of the nation's waters, end quote. Despite the 1987 Act's refocus on ambient conditions, regulatory focus has remained on point sources, those discharging pollutants into receiving waters at specific locations. However, there are pollutants entering water bodies from diffuse sources and hydrologic alterations that cause non-point source pollution. Applying NPDES effluent-based standard criteria to non-point source dischargers has been highly problematic and ineffective. There are consequences from applying effluent-based rather than ambient-based standards to diffuse non-point source pollution discharges. First, regulatory efforts to measure and communicate water quality accomplishments were often described in terms of compliance with wastewater permit conditions rather than the condition of the receiving water. Second, effluent standards should apply only to point sources rather than to all sources of a pollutant or other forms of pollution. Pollutants from non-point sources derived from diffuse and hard-to-monitor origins such as agricultural, silvicultural, and construction activities largely escaped oversight. Third, attention to chemical pollutants measured in discharge water came to dominate water quality policy, while physical and biological determinants of the ambient conditions were only infrequently considered. In many places, particularly non-urban areas, the condition of a water body depends on more than the loads of specific pollutants from sources discharging under an NPDES permit. For example, changes in the hydrologic regime associated with residential and industrial development activities can destabilize stream banks, increase loads of sediment and nutrients, eliminate key aquatic species, or otherwise change the aquatic ecosystem. Biological, hydrological, and physical changes to a water body that do not fit the definition of a pollutant were included in the 1987 Act's definition of pollution. The CWA defines the two forms because both are important contributors to a water body's assessment. The Clean Water Act defines pollutant as dredge spoil, solid waste, incinerator residue, biological materials, radioactive materials, heat, wrecked or discarded equipment, rock, salt, cellar dirt, and industrial, municipal, and agricultural waste discharged into water. The Clean Water Act defines pollution as the man-made or man-induced alteration of the chemical, physical, biological, and radiological integrity of water. In the Act, pollution includes pollutants as well as other stressors such as habitat destruction and hydrologic modification. The CWA objective of whole ecosystem integrity cannot be met or even approached by application of effluent-based criteria for TMDL water quality standards. Effluent limitations are the primary mechanism for controlling discharges of pollutants to receiving waters in NPDES permits. When developing effluent limits, a permit writer must consider the available technology, that is, technology-based effluent limits, or TMDLs, and what would be protective of the water quality standards of the receiving water, that is, water quality-based effluent limits, or WQBELs. Effluent limitation guidelines and standards are established by EPA for different non-municipal, that is, industrial categories. These guidelines are developed based on the degree of pollutant reduction obtainable by an industrial category through the application of pollutant control technologies. The definition of ambient is surrounding or enclosing. This means that to comply with the CWA's objective of restoring the chemical, physical, and biological integrity of water bodies, the entire local ecosystem needs to be characterized, not just chemical ions and molecules. More on this point when I discuss water body assessment in detail. Ambient conditions apply to specific stream and river reaches and tributary subbasins called assessment units, in addition to the whole river network. Because TMDLs are supposed to correct the status of an assessment unit ecosystem in its defined area, the maximum loads should be based on achieving desired future ambient conditions as required by the 1987 Act. Equally important, setting standards and objectives based on desired ambient future conditions applies to aquatic Superfund sites under the Comprehensive Environmental Response, Compensation and Liability Act, CERCLA, and other water quality laws. TMDL allocations include the effluent-based standards for point source discharges, as well as those that contribute to the TMDL and are allocated with non-point sources in the assessment unit. Compliance with this section requires states to identify waters not meeting ambient water quality standards, define the pollutants and the sources responsible for the degradation, establish total maximum daily loads necessary to achieve those standards, and allocate daily foot loads to all sources within the assessment area. Therefore, for each imposed assessment unit, the state must identify the amount by which both point and non-point sources need to be reduced to meet ambient water quality standards. Other degradation factors that do not fit the pollutant definition, such as changes of habitat, flow alteration, channelization, and modification or loss of riparian habitat, may be considered as reasons for not meeting standards. The assessment process needs to be explained in terms of regulatory science, biology, ecology, and statistics. I'll do this using words, no equations or mathematics. The TMDL process depends on observations and measurements. Therefore, it is significantly influenced by the setting of water quality standards. Water quality standards consist of two parts. 1. A specific use appropriate to the assessment unit. This is called a designated use. 2. A measurable criterion to establish whether the designated use is being achieved. Oregon refers to designated uses in statute and regulation as beneficial uses. I use designated uses because it's a more neutral term. A designated use describes the goal of the water quality standard. The Natural Resource Council's TMDL review found that stating a designated use with terms such as fishable and swimmable are too broad to be useful. It is better to distinguish between, for example, full body contact with water by swimming and partial contact while boating or wading when fishing. A better description for a designated use of human contact recreation should protect humans from microbial pathogens while swimming, wading, or boating. Other designated uses should be defined to protect humans, wildlife, and livestock from harmful substances in water and fish tissues. Specific aquatic life designated uses should be designed to protect fish and shellfish. A designated use is qualitatively described in a document and should be as specific as possible. Thus, more detail than, quote, recreational support, end quote, or aquatic life support is needed. The general fishable and swimmable goals of the Clean Water Act are the beginning rather than the end of an appropriate use designation. For example, a sufficiently detailed designated use might distinguish between beach use, primary water contact recreation, and secondary water contact recreation. Similarly, rather than stating that the assessment unit needs to be fishable, the designated use will ideally describe whether the water body is expected to support a desired fish population, for example, salmon, trout, or bass, and the benthic macroinvertebrates or other biological communities necessary to support them. Though small headwater streams may have aesthetic value, they may not have the ability to support being fishable or swimmable. However, Their condition has an influence on the ability of a downstream area to achieve a particular designated use. Here, the designated use for the smaller assessment unit may be defined in terms of the designated use of the larger downstream assessment unit. A water quality standard should include a measurable indicator supporting the designated use. One set of criteria could include acceptable constituent measurements of dissolved oxygen, temperature, nutrients, pH, suspended sediments, and other water constituents. One constituent alone is insufficient, as animals living in the water, as well as human full-body contact, are affected by the entire composition of constituents rather than a single one. Additional criteria could include measures of the algal community, benthic macroinvertebrates, or pollutant concentrations in fish tissues. More general criteria applicable at the basin or watershed level include sources of pollution other than pollutants. These criteria might include flow timing and pattern, a hydrologic criteria, abundance of non-indigenous plants and animals, a biological criterion, and some measures of channel modification such as a decrease in sinuosity, geomorphic criteria. Surrogate variables such as chemical concentrations often are selected for use as criteria because they are easy to measure. Although the surrogate may have this appealing attribute, its usefulness can be limited if it cannot be logically related to a designated use. For example, chlorophyll A has been chosen as a biocriterion in some states because it is a surrogate for aesthetic condition or as the sole measure of the status of a large aquatic ecosystem. Biological criteria should be used along with physical and chemical criteria to determine whether an assessment unit is meeting its designated use. The most weight should be placed on biological criteria because they are more closely related to the long-term ambient conditions than our chemical measurements. However, guiding management actions to achieve water quality goals based on biological criteria depend on adapting appropriate modeling efforts. All chemical criteria and some biological criteria should be defined in terms of their magnitude, frequency, and duration. Magnitude should be the 95% confidence limits of natural variability or of a constituent at all seasons and typical water levels. Frequency should be expressed in terms of the number of allowed confidence limit exceedances in a specified period. Frequency should be, duration should be how long this criterion exceeds the confidence limit. Establishing these three dimensions of the criterion is crucial for successfully developing water quality standards and subsequent TMDLs if justified. Water quality standards need the most reasonably obtainable monitoring data. That is, there should be a sufficiently large number of samples to characterize natural variability and excursions beyond that. Unfortunately, Oregon and most states don't have enough spatial and temporal data to limit data uncertainties. Regulatory decisions too often take longer than they should. Because natural ecosystems are highly complex and variable, our attempts to understand how human activities affect them is incomplete, and there is never a single right answer. The extreme result is paralysis by analysis. Ambient-based water quality management requires asking whether the designated use is being attained. If it's not, the reasons need to be identified and decisions made on how the situation can be remedied. These issues, referencing specific criteria, can't be answered with certainty. And it's because of this uncertainty that most management programs make serious mistakes. Uncertainties are a fact of life. The better we understand them, the better are the decisions we make. So let's look at how we understand uncertainties and use our knowledge to make more technically sound, informed, and legally defensible decisions. We know uncertainties are everywhere because Benjamin Franklin pointed out that in this world, nothing can be said to be certain except death and taxes. Our efforts to explain the world is often filled with uncertainties, and this is the major reason why environmental regulations is both difficult for regulators and highly contentious with the regulated public and other stakeholders. Statistics is the science of uncertainties, and it offers a coherent and mathematically robust approach to identifying and quantifying sources of uncertainties. In common use, uncertainty is expressed as variability. Total variability is the sum of variability due to measurement, using a model of how the world works, and parameters that control the measurement and model variability. For all environmental science, there are three components of uncertainties. First, measurement variability in the field and laboratory. Second, model variability in explaining data. Third, parameter variability in describing measurements and models, for example, means and variance. Although real-world systems may in principle be partly deterministic, our information is incomplete at each stage of observation, summarization, and inference, and so our understanding is clouded by uncertainty. When the dynamics of the system are nonlinear, the process can exhibit chaos even though the theory is based on deterministic dynamical systems. Accepting the universal presence of inherent natural variability allows us to view environmental regulations as scientific understanding in the presence of uncertainty. All environmental observations and measurements are tied to a specific location and time, and all data include some error. In addition to obvious errors with measuring, manipulating, and storing, data often have other errors such as sampling water at discrete places at specific times in what is really a continuous system. What I mean by this is that all environmental data, and especially data from aquatic ecosystems, provide a series of individual pictures and is similar to understanding a movie by watching a series of still shots. Also, in context of environmental regulatory science, spatiotemporal data are rarely sampled at places or times optimal for answering a specific regulatory problem, such as assessing water quality in a river network to develop and allocate a TMDL. All scientific attempts to explain the world in which we live All science attempts to explain the world in which we live, but our world is very complex. A model is a simplification of some well-chosen aspects of the world, where the level of complexity often depends on the question being asked. Pragmatically, the goal of a model is to predict, while scientists want to incorporate their understanding of how the world works into their models. Models that are scientifically meaningful, that predict well, and are conceptually simple are preferred. While it's easy to elevate simplicity over the other two factors, doing so can lead to incorrect results. For example, a statistical model based on correlational associations might be simpler than a model based on ecological theory. The way to bridge this divide between simplicity and scientific soundness is to use appropriate statistical relationships to characterize it. Nearby measurements tend to be more alike. This is especially important with geochemical data for assessing a designated use's status. Chemical concentrations will be more similar even in flowing waters when samples are collected in the same general area than are samples collected from distant sites. Streams and rivers in urban areas and in locations where industrial activities are adjacent to the waters tend to have data collected and analyzed more frequently than are rural or headwater streams. When predicting values in areas not sampled, the relative distance to sample data should be included in the calculation. This parameter, location relative to other sample locations, has a strong influence on basin-wide water quality assessments that result in TMDL development. Variability within a population of organisms can be very high. People are a good example of this. The concentration of pollutants in surface and groundwaters, sediments, and soils can also display very high variability. Analyzing observational data requires different statistical models than do experimental data found in medicine and agriculture. Consider the controversies involving greater sage grouse, spotted owls, and Pacific salmonid stocks. Survey technicians may look or listen in habitats known to be used by these species they want to count, but in these situations the aphorism that absence of evidence is not evidence of absence must be taken into consideration when selecting models to fit these data. Anyone who fishes or hunts can confirm that not finding an animal when you're in the area does not mean they're absent, only that you did not observe them. Geochemical data are also observational. No one has conducted experiments on the chemical concentrations in a river, grassland soils, or ore bodies. Whether the data represent chemical, physical, or biological components of an aquatic ecosystem, they are observational, and the statistical models found in spreadsheets and basic stat courses in university science and business programs cannot produce valid results because they were designed for experimental data. This means that frequentist hypothesis testing models can be used in only very limited situations with environmental data. With the regulatory and science background I presented, it's time to look at how water bodies are assessed for compliance with the CWA goal. I'm presenting Oregon's approach because I'm much more familiar with this than with that of other states. Oregon's environmental regulator says they apply the NRC requirements of using ambient monitoring as the basis for determining whether waters are placed on the CWA Section 305B preliminary list or on the action list. However, Oregon's process is described in Section 3.3.4 of the document titled Methodology for Oregon's 2018 Water Quality Report and List of Water Quality Limited Waters. Unfortunately, it specifies statistical approaches and models that do not fit geochemical data such as surface water constituent concentrations. Nor does it include specifics for assessing the physical and biological status of the assessment unit. The agency's focus only on pollution. On pollutants, no pollution factors are considered. I'll explain in detail why these are wrong because it's important that you understand this. A fundamental rule in statistical data analysis is to fit the model to the data, not the data to the model. The first sentence of the DEQ description reads, DEQ shall use a statistical hypothesis testing approach, binomial test, to derive a critical number of sample excursions that scales with the number of representative samples to evaluate beneficial use attainment status of water bodies. While I understand all the words, I don't understand the entire sentence. This doesn't matter as there is much wrong with it. So I start with the basics and work forward from there. Oregon uses some EPA-recommended assessment categories, specifically Category 2, which says that available data and information indicate that some designated uses are supported and the water quality standard is attained. And there's Category 3D, that says there are insufficient data to determine use support because numeric criteria are less than quantitation limits. This means the presence and concentration cannot be measured. The data are censored, that is, unknown. This needs explanation. Censored geochemical data are those below the analytical chemical laboratory's method detection limit. There are two interpretations of censored data. One, the lab cannot determine if the constituent is present. Two, if it is present, the concentration is too low to be absolutely quantified. It is usually toxic metals and organic compounds that are censored when the lab looks for them. The problem with downgrading an assessment unit to Category 3D because the lab cannot find any toxic metals or organic compounds for the state to compare with the regulator's fixed maximum values does not make scientific or policy sense. If there is neither no toxin present or the presence is too low to quantify, the water certainly satisfies all beneficial uses concerning that chemical toxin. The data are not insufficient, they are sufficiently clear. There is no reason for any designated use to be adversely affected by the absence or undetectable quantities in the water. As I mentioned before, the hypothesis testing approach was developed for use in analyzing experimental results. It does not apply to geochemical data because a set of constituent concentrations cannot be assigned to null or alternative hypotheses. In addition, the hypothesis testing approach cannot be applied to diffuse pollution elements such as habitat loss, channel alteration, or biota community structure and function in the assessment unit. I need to spend a few minutes with the binomial test. You're not statisticians, and I'm not going to train you as statisticians, but you need to understand why this specific test will not provide correct or useful results. Binomial literally means two numbers. In probability theory and statistics, a binomial distribution is a discrete distribution of counts of the number of successes in a sequence of independent experiments, each asking a yes-no question, and each with its own limited outcome of success or failure, yes or no, true or false. Chemical concentrations are continuous, not discrete variables. Neither a concentration nor a set of concentrations can be characterized into yes-no, success-failure, or true-false dichotomies. The statistical binomial test is an exact test of the statistical significance of deviation from a theoretical expected distribution of observations into two categories. DEQ is looking for deviations from a binomial distribution for continuous concentration values using a test designed for discrete values or counts. Perhaps the agency got this idea from a common ecological test applied to biological population surveys. An example will help you understand this. You're doing periodic surveys of summer steelhead trout redds, or nests, in several reaches of the upper Deschutes River in the second week of June, and you have 10 years of red counts for each reach. When you go out this year, you have an idea of how many reds you expect to find in each survey area because you know the mean number of reds in each reach over the 10 years. You can compare your observed red counts to the expected red counts by calculating how well the data are fit by the chi-square probability distribution. The chi-square tests the hypothesis that the observed counts are no different from the expected counts. This applies to counts, discrete numbers, not to a binomial distribution for discrete data, and certainly not for continuous chemical concentration data. The rest of the methods described in section 3.3.4 are also based on trying to fit continuous chemical concentration values into statistical models not designed for this type of data. Other states likely have their own approach for applying ambient conditions to assessing water bodies, but I'm not as familiar with them as I am with Oregon's. Regardless, I want you to have an understanding of one more bit of statistics, the expected value. There are three types of expected values called means in probability and statistics. First, the arithmetic mean. This is the central value in a distribution of continuous variables such as geochemical concentrations. Second, the geometric mean. This is the central value in a distribution of positive numbers that represent rates such as investment returns or rate of growth. Third, the harmonic mean, which is the central value of a set of numbers defined in relation to a unit such as speed, which is distance per unit of time. Oregon uses the geometric mean for the average of water constituent concentrations when the arithmetic mean should be used. When states or the EPA set a single maximum concentration limit for a pollutant in all waters of the state, they ignore natural variability. In Oregon, for example, having the same maximum applied to Crater Lake, Lake Oswego, the Malheur River, and the Willamette River ignores how different each is from all other water bodies. About 20 years ago, the EPA dropped the maximum level of arsenic in drinking waters from 50 micrograms per liter that had stood since 1948 to 10 micrograms per liter. Yet there are many areas in the U.S. that have naturally high arsenic concentrations, and no one dies from them. According to a 2019 survey by the U.S. Geological Survey and the Centers for Disease Control, based on studies of more than 20,000 domestic wells across the country, about 2.1 million people drink water that is naturally much higher than 10 micrograms per liter in arsenic, and all from natural sources, not from industry. In Oregon, counties range from 0 to 5,000 people drinking water with greater than 10 micrograms per liter. Clark County in southern Nevada has more than 5,000 people drinking water with arsenic levels greater than 10 micrograms per liter in some domestic water wells. States should transition to using biological data to set water quality standards. Documenting how a regulatory process produces incorrect results is insufficient. An alternative needs to be provided because the assessment process can put water bodies on the TMDL implementation list when they are not impaired. Ambient water quality considers the overall physical, chemical, and biological condition of the aquatic ecosystem being assessed. Local biota represent ambient water quality much better than do fluctuating chemical constituent concentrations because the biota are there as water quality varies within its natural range. Fish are not found in every assessment unit, in all reaches of those they do inhabit, and they move around a lot, so they're not suitable indicators of ambient water quality. But benthic macroinvertebrates, the animals living on the water body substrate, are found almost everywhere along the river network from the highest first-order streams to the mouth of the river, and in almost all ponds, lakes, and reservoirs. Benthic macroinvertebrates include the juvenile stages of insects, arachnids such as mites, snails, freshwater mussels, worms, and other small animals. While most individual species are present for only one generation per year, the community composition remains relatively stable because their food resources are relatively stable and characteristic of the reach's position within the river continuum. All biota, from microbes such as bacteria and fungi through plants and animals, are classified into taxonomic groups based on common anatomical characteristics. Benthic macroinvertebrates are easily identified into the three taxonomic levels of class, order, and family. And each family has been identified to one of 5 feeding categories: shredders, filterers, gatherers, grazers, and predators. Some stream ecologists combine filterers and gatherers into the category of collectors. Shredders feed on large particles such as leaves and twigs, the organic material common to headwater reaches that are covered by overhanging riparian vegetation. Filterers capture fine particles of organic matter from the water column with little nets. Gatherers collect loose fine organic particles on the stream bed, and grazers feed on algae and moss growing on rocks on the bottom substrate. Predators feed on other animals. Almost all benthic macroinvertebrate families are easily placed in a feeding group by their predominant feeding strategy. This information has been published and is widely available. This makes them ideal for monitoring ambient water quality. This diagram of the river continuum concept illustrates how the river network and its aquatic biota change from the headwaters to the mouth. Notice that the relative proportion of feeding strategies changes longitudinally as the food resources change. The term aquatic life has no consensus definition, so I'm defining it as the range of natural variability in the relative proportion of taxa in each feeding group. This range of variability will differ by basin and by location within the river network. A combination of geomorphic, hydrologic, and chemical explanatory variables can be analyzed for their contribution to the observed relative proportion of feeding groups. There are two specific questions the data analyses are to answer. First, what is the normal range of variability of the composition of feeding group proportions in assessment unit based on available data? Second, what geomorphic, hydrologic, and chemical variables explain the observed patterns? When analyses yield results beyond the normal range, it is necessary to examine the individual explanatory variables to determine which have changed and identify the reason for that change. This is another perspective on benthic macroinvertebrate feeding group proportions in a river network. Notice how the composition of the feeding groups change along the river continuum as the food resources change. The proportions remain relatively constant, but the specific taxa change by location and season. What is valuable in assessing ambient water quality conditions is the amount of variability, uncertainties, in each sampling location. I'll explain how to use this variability to assess the reach's compliance with the CWA objectives. It is worth repeating the two questions for assessing the role of biological communities in water quality status. 1. What are the differences from one collection time to the next? And 2, what environmental variables explain the observed pattern of biological community composition? There are many measures of association, commonly called similarity measures or coefficients, that might be applied to biological communities. For setting water quality standards and detecting changes that adversely impact the designated use, only a few measures will work. Since only 5 feeding groups are commonly used, differences in relative proportions among these groups and the environmental explanatory variables that affect them are specific to each stream reach and designated use. The numbers of taxa collected will always vary because of both natural and anthropogenic causes. Not all possible feeding strategies are represented in the community composition because some, such as parasites and omnivores, are both relatively rare and infrequently identified and counted. Relative proportions of taxa in each major feeding group are less variable than the counts themselves and contain all the important information about the biologic community's function. Because feeding groups reflect energy processing and nutrient spiraling along the stream network, the ecological interpretation based on proportions of taxa is relatively straightforward. The numbers and proportions of individuals at any given collection event will vary according to their life cycles, but the proportion of taxa will be more constant because feeding groups reflect the range of available niches in the collection location. That is, not all individuals actually present on the sampled substrate will be collected, and those collected cannot be assumed to represent their proportions in the entire benthic macroinvertebrate population. But all habitats will have representatives feeding on available particles, organic matter, and there will be predators feeding upon the other animals. The concept of compositional data analysis originates from work by a French mathematician in 1866. In 1879, the British mathematician and biometrician Carl Pearson argued that only proportions between 0 and 1 will produce useful results. The first statistical theory and models for analyzing compositional data were published by John Aitchison in 1986. In addition to economic and other public statistics released by governments, compositional data analysis was quickly adopted by mathematical geochemists to examine the chemical composition of rocks, including ores, and soils. Much more recently, it has been applied to pollution analyses and biological and ecological data. Before determining whether a permitted activity has adverse impacts on a specific designated use, it's necessary to quantify inherent natural variability. Regression analysis is the class of statistical models that measures the relationship between the value of a response variable and the measured range of explanatory variables. For setting water quality standards and evaluating compliance with them using benthic macroinvertebrate feeding groups, compositional data are the response variable in regression models. Regression equations can be used to forecast future values of the response variable, assuming that the relationships to the explanatory variables remains constant. Time series analyses are also usefully applied to compositional data. Several types of data plots or figures can describe compositional data and their relationships to explanatory variables so that we better understand the ecosystem. Of these, ternary diagrams illustrate data distributions useful for regulatory decision-making. These are not commonly encountered in regulatory compliance reports. A ternary plot is explained. Ternary diagrams plot the position of a point based on 3 or more variables in two-dimensional space, such as a printed page. They are common in geology and geochemistry, but much less common in displaying environmental data. This is a ternary diagram showing the relative proportions of feeding groups in all pairwise comparisons. The 95% confidence limits for each pair is shown in red. The upper and lower halves of the matrix are mirror images, so pick one and compare within that set of plots. I won't go into a detailed explanation now, but you can see that over the available data set, the variability among temporal samples is well defined, so any future collections that plot outside the limits indicate a close look at explanatory variables is needed. One way to look at several explanatory variables and how benthic macroinvertebrate communities respond to them is illustrated in this ternary plot. This plot shows how biota are affected by all chemical constituents in their environment rather than a single one. Applying this type of analytical model to ambient-based assessments produces insights that help inform technically sound and legally defensible policies and regulations. Oregon's Department of Environmental Quality, and probably most other states, follow these steps based on the data they have available. Characterize the water quality conditions and inform assessment status. Develop and calculate TMDL models. fill in existing data gaps, set load limits for listed water quality parameters, help determine source controls needed to meet load limits. I've explained how benthic macroinvertebrate feeding group composition provides a robust basis for characterizing ambient water quality and variability in all water bodies. According to one of their websites, the EPA's direct role is limited to listing impaired waters and planning their recovery. The implementation of recovery actions, measurement of their efficacy, and declaration of recovery are primarily up to the states. Again, as an aquatic ecologist and fluviogeomorphologist with decades of experience, I've learned that each stream, drainage basin, and river network is unique. Applying fixed deterministic differential equation models that are supposed to fit all water bodies in the county or in the country will not yield useful results. When an assessment indicates the need for remedial TMDL, a custom-fit approach based on established ecological, hydrological, and geomorphic theory should be developed and implemented. The theory and practices for river restoration are well established because they work with the river's natural tendencies and result in self-sustaining and fully functional aquatic ecosystems. This more involved process is needed in today's megadrought conditions. As of late spring 2020, the upper Missouri River is totally dry for a long distance in eastern Montana and western South Dakota, a situation that's not existed for 1,200 years. Replacing the past fixed process of applying chemical effluent concentrations with biotic and ambient conditions is not only justified today, but necessary to continue providing clean water for all designated uses and all water bodies. What I want you to remember are, it's all about the data. Ambient-based water quality standards include pollution as well as pollutants. For TMDL assessment units, Biota provide more technically sound and legally defensible decisions than do chemical data, especially effluent-based standards. When it's important how environmental data are analyzed and interpreted to be technically sound and legally defensible to avoid and resolve environmental concerns, contact me. Here's information how you can get in touch with me.